So, good afternoon to all of you. And I hope all of you might have read the new circular that has been issued by the Karnataka University. ಸಿಕ್ಕಿದೆಯಾ ನಿಮಗೆ? ಹಲೋ ಹಲೋ ಹೇಳಿ ನಿಮಗೆ ಗೊತ್ತಿದೆಯಾ ಇಲ್ವಾ ಹೊಸ ಚೇಂಜಸ್ ಇನ್ ಎಕ್ಸಾಮ್ ಪ್ಯಾಟರ್ನ್ ಅಂಡ್ ಆಲ್. So for both odd and even semester you will have only MCQ type questions, that is multiple choice question, and question paper will be of 40 marks question, 40 questions, each question carrying 2 marks. First there will be test for your present semester, and after that in the end there will be test for the previous semester. First each semester dane ಮಾಡ್ತಾ ಇದ್ದಾರೆ ನಿಮಗೆ. Probably practicals will start from August 1st up to 14th ಒಳಗಡೆ we have to complete all the practicals. Before that the theory part we should complete. Then daily there will be 3 examination, 9 to 11 and then daily 3 exams ಇರುತ್ತೆ, but ಒಂದು ಕ್ಲಾಸಿಗೆ ಒಂದೇ ಎಕ್ಸಾಮ್ ಇರುತ್ತೆ. So like that they have made it, and prepare for that. Okay, now we will move on to the topic. We will continue our discussion on the chromosomes. So the chromosomes, we have in the last class we have tried to understand about the chromosome structure and all those things. Even this also I think we have discussed the chromosome structure, how they the protein core, linker DNA, and then ultimately how by coiling and many times multiple coilings, what you call the solenoid coiling, they will attain the shape of a chromosome. So this is how the chromosome ultrastructure appears. So again I will tell you, see here in the first photograph you can see the histone core, and this is the DNA which wraps, usually it wraps 1.75 per turns it makes. Then these beads are joined by what you call the linker DNA, and this appears like beads on strings on the nucleosome. So the nucleosome, when you observe, I don't know whether we have observed in the earlier or this. Yeah, here you can see the beads on string. That is the nucleosome how it appears under the electron microscope. If you further analyze, this is how it has been arranged. So the nucleosome includes around approximately 200 nucleotide base pairs, and the linkage is done with the help of this linker DNA. This yellow structure is the histone core. So then suppose if you hydrolyze this, if with the help of an enzyme, that is the nuclease enzyme, which will first digest this linker DNA and makes into nucleosome core particles. See this, each nucleosome core particle is 11 nanometer in diameter, remember, see, and it has 1.75 turns of DNA, which consists of around 146 nucleotide base pairs, double helical. This is when you observe. Now suppose if you expose this nucleosome core particle with high concentration of the salt, again it breaks down into a 146 base pair DNA double helix, which makes 1.75 turns anti-clockwise, and then there will be an octameric histone core. It is having octameric structure, and the histone consists of H2A, 2 molecules of H2A, 2 molecules of H2B, 2 molecules of H3, and 2 molecules of H4. This is the fine and ultra structure of a nucleosome, which we have already discussed. That again I am repeating. ಅರ್ಥ ಆಯ್ತಾ ಇಲ್ವಾ ಇದು ಹೇಳಿ, because it's most important even from examination point of view. Yes,
Yes, sir.
okay, if you have understood then I will move it. So this nucleosome core particle, see, it looks like that. See here, this is the different the histones. So there's one, the H2B, H2A, H2B, H3, and H4. And see, this is the DNA, what you call the linker DNA. And surrounding that, there's a DNA. And this is the one which joins with the next one. So that's why this and this portion you call it as linker DNA. Now, after studying the uh, ultrastructure of the chromosome, we should know something about the sex chromosome. So this sex chromosomes, uh, actually when the ideogram is placed, I have told you there are two, they are kept separately and they are known as X and Y chromosomes. So this also you know, which I would have discussed earlier. Now what is important for us is there any variation in structure of chromosome? Suppose if there is a variation in the structure of chromosome, what can happen? What are the consequences? Then what type of variation can occur in a chromosome, either naturally most of the time or sometimes these variations can be induced also. So this we have to study the variation in structure of the chromosome. So and they are also called as chromosomal aberration. Chromosomal aberration. So in all the somatic cells, there will be somatic and gametic chromosomes. Somatic chromosomes are always in pairs 2n, and in the gametic 2n, the chromosomes number of a species that remains constant. Suppose to take the example of our own human beings, we have how many chromosomes? We have 23 pairs. That is actually in a non-dividing cell or in a interface, the cell which is under interface, they are, you can have diploid chromosomes, diploid, they are double set. So that's why they are called as 2n. But when the cell is in a dividing state, then in the division you can have a haploid condition or what you call n, especially during the sexual reproduction you can expect. But this number remains same. Either it may be 23 pairs or 23. Uh, this constant chromosome number is because of the extremely precise cell division, both mitotic as well as meiotic cell division, which are very precise that they ensure the chromosome number constant. Because in each step there's a proofreading mechanism, and if at all any small error occurs, in majority of the time they are rectified and corrected. So that's why the chromosome number remains constant. Somatic cells of the diploid species contain two copies of each chromosome, and these two copies, they are homologous chromosomes. These two copies of the same type of chromosome, that is what we call homologous chromosome. And that's why even their gametes therefore contain, but whereas the gametes contain only one copy of each chromosome. Gametes chromosome number is only one. It is because when there are two gametes, male gamete and female gamete. For example, if you take the even human being, the male gamete will contain one set of chromosome. Female egg will also have one set of chromosome. That is 23 in the maternal side and 23 from the paternal side. When they fuse and when the zygote is formed, diploid is restored. That means the zygote will have 23 pairs of chromosome. So that's why you know in meiotic division the chromosome number is reduced to half, if you are able to recollect what you have studied in your PUC or in high schools. So this is very essential in maintaining the constant number of chromosome from generation to generation. Suppose if the reduction doesn't happen, means if the reduction do not happen, what will happen? Male gamete will have 23 pairs, female gamete will have 23 pairs, the zygote will have 46 pairs. So from each generation the chromosome number goes on doubling. So but it never happens because the gametes will have only haploid number of chromosomes. In the during the gametogenesis, during the gametogenesis there's a meiosis. In the meiosis the chromosome number is reduced to half. So each chromosome of a genome contains a definite number, and even not only the chromosome, each chromosome of a genome contains definite number and kind of genes, and all these genes are arranged in a definite sequence. They are arranged in a definite sequence and they contain a definite number um, and kinds of gene. Now, with this background, with this information, let's try to study the chromosomal aberration, the variation. Sometimes due to mutation or spontaneous variation, we don't know why, without any known causal factor, we can say that it is spontaneous. Or sometimes it is because of the mutation. Variation in the chromosome number or the structure of the chromosome arises in nature. See, chromosome number should be constant, and even the variation in the chromosomal structure, it should not happen because in the, you know that there's a definite number and kinds of genes which are arranged in a definite sequence in a chromosome, on a chromosome, remember. But sometimes either the variation in the chromosome number or the structure do arise in nature, and this variation either in the chromosome number or structure we call it as chromosomal aberrations. And these chromosomal aberrations may be grouped under two broad classes. One is the structural aberration and another one the numerical variation. Structural aberration with respect to the structure of the chromosome, with respect to the kind and sequence of arrangement of the genes, and in the second one, the numerical, it is the change in the chromosome number, otherwise which would have been constant. Now, first let's try to understand the structural aberrations of the chromosome. The chromosomal structural variation is the result from chromosomal breakage. Sometimes there will be breakage in the chromosome. Chromosomal, as a result, the structure of the chromosome varies. Some kinds of genes may not be present, or the sequence of the arrangement of the genes may change. These are because of the structural aberrations. And this, when the chromosomes are broken, they tend to rejoin. This is a very, very interesting thing. See, while explaining the telomere, I have told you at the terminal end of the chromosomes, they are coated or plastered with a certain kind of protein, and as a result of that, the chromosomes will maintain its stability and uniqueness. Suppose if that protein coats are not there on the telomere, the chromosomes become very, very sticky. But here what happens? The chromosome has been broken. The broken ends of the chromosomes are very, very sticky. Being sticky in nature, they tend to rejoin. Suppose if there are more than one break, the rejoining becomes random. Not necessarily with the correct ends. So as a result, what has happened? The sequence of the genes have changed. This results in the structural changes in the chromosome. So now you may ask how these chromosomal breakages can happen. It can happen because of X-rays, continuous exposure to X-ray. It may cause the structural aberrations, or various chemicals like mustard gas or radiation, and in some cases it can happen spontaneously without any known reason. So in nature there can be four common types of structural aberration. There can be four common types of structural aberration. They are called as deletion or deficiency, second one duplication or repeat, third one is inversion, and the fourth one is translocation. So in generally, if you classify these structural aberrations of a chromosome, broadly you can categorize them under these four titles. Now, for the better understanding of each of the variation, let's consider a chromosome on which the genes are arranged in this fashion. A chromosome with genes in alphabetical order. First there will be A gene, then B, then C, D, E, F, R, F, G, H, I, like that. This is the arrangement of the genes in an alphabetical order on a normal chromosome. Now let's try. The first one, the deletion. What has happened is a part of the chromosome has been removed. A part of the chromosome has been removed. As a result, this chromosome has become shortened because here only we have the genes ABC and GHI. This DEF, this piece is missing. See, DEF, this piece is missing. So those genes are not there. Chromosome has shrunken, it has become small in size, shorter in size. It is because of the loss of some genes which are arranged. This type is known as deletion. Second one is the duplication. A part of the chromosome is duplicated. An additional piece has been joined. See how A, B, C, D, E, F. After that, again, three more genes which have been lost from here, D, E, F, have joined here. See, this is the gene which has been missing from this deletion that has joined the other chromosome in a different order. A, B, C, D, E, F, A, B, C, D, E, F, and there are two genes of the same type, C, D, E, F and D, E, F. These are the genes of the same type. Where here there is a loss of those genes, here those genes are added. Then it continues GSI. This is what you call duplication. Here see these genes DEF genes have been duplicated. That's why it is called as DEF. Then the third type is inversion. The part of the chromosome has been reinserted in a reverse order. A part of the chromosome has been reinserted in a reverse order. Otherwise, you know, ABC, DEF, GHI. See, this gene, the E gene, it has misread sequence and it has been reinserted in a reverse manner. Actually, E should come after D, but it has come before that. This gene has come before this gene. So, this is what you call inversion. Then translocation is nothing but the ring. The ends of the chromosomes are joined together, make a ring. The ends of the chromosomes, because of the breakage, the ends have become very sticky and they have joined together. What has done? A joins with I and forming the chromosome instead of rod-like structure, it makes it a ring-like structure. Even translocation, sometime it may be parts of two non-homologous chromosomes are joined. If there are two normal chromosomes, if both of them show the aberration, then if there are two chromosomes, see one chromosome ABCDFGHI and another chromosome UVWXYZ. These are the arrangement of genes on these two chromosomes. But the breakage has have occurred in both the chromosomes, that is ABCD as ABCDFGH as well as UVWXYZ. Now what has happened? A piece of this has joined with this chromosome and a small piece of genes has joined here. See, it has become ABCDF XYZ. This piece has lost from here and joined with this. Then here you have UVW, but the GHI, this portion has joined here. So this is what you exchange of the pieces of the chromosome between two non-homologous chromosomes. Actually, this exchange or swapping takes place in the homologous chromosome during the uh, especially in genetics that you will translocation and you can study that where the exchange of swapping of the genes takes place between the two homologous chromosomes. That's must there. But here this occurs between the two chromosomes, two non-homologous chromosomes. See, this is A, B, C, D, E, F, G, H, I. Deletion A, B, C, E, F, G, H, I. As one is missing. See here in duplication A, B, C and a small piece from this has also entered and the chromosome has increased in length. In inversion, you can see it has been placed reversely. Translocation, this is ABCDFGH, this is MNO, like that MNOPQR. See, this piece to this, a small piece of this has attached, and to this, a small piece of this has attached. So this is an unequal crossing over, duplication of two copies. See, this is the chromosome, some chromosome, they are named as 16A and 16A double. Unequal crossing over, see, 16A, 16A, again there's an exchange between that. Three copies, like that it happens. And inversion consists of two breaks, I should say that, in one chromosome. For the inversion to happen, there should be two breaks in one chromosome. The area between the break is inverted, turn around and then reinserted in the break. That unite with the rest of the chromosome. This is how it has happened. See in the E. If the inverted area includes centromere, suppose if the inversion happens within the centromere, then it is called as pericentric inversion. So these are the chromosomal aberration with respect to their structure. That means with respect to the arrangement of the genes. This is what we call one chromosomal aberration. Other one is variation in the chromosome number. Even you can call it as chromosomal abnormality. This chromosomal abnormality, that is with respect to the chromosome number is concerned. Otherwise in normal cell, all of you know it is constant. But sometimes there will be abnormalities. Basically one can expect three different types of abnormalities. One is polyploidy. Poly means many, ploidy means sets. Having more than two sets of chromosomes. Usually in a set there are two chromosomes, but here there will be one extra, more than two sets of chromosomes. Like that, that is what you call polyploidy. There's also one more, aneuploidy. Having an extra copy of a single chromosome. One extra copy of a single chromosome. Or missing a copy. Sometimes there can be extra copy, sometimes there may be missing of one copy. So this is what you call aneuploidy. Then mixoploidy, having a cell line with different chromosomal constitution. Remember, in a cell, if there are 23 chromosomes, all these chromosomes have some similarity with respect to their chromosomal constitution. But in mixoploidy, this gets changed. For example, in mosaics, that is mosaics derived from a single zygote. Generally, for the formation of zygote, there must be fusion of two gametes, that's the male and female gamete. But in mosaics, it is derived from the single zygote, that is mixoploidy. Or if you have remembered what you have studied in BSc 1, the chimeras, which belong to subclass holocephaly under the fishes. Chimeras. These chimeras are derived from the fusion of two different embryos. This is a very interesting thing. Actually, zygote developed by the fusion of two gametes. But here there is no question of zygote formation. Instead of that, there will be two different embryos and they fuse. Now, with this background, let's study about the variation in the chromosome numbers. Organisms with one complete set of chromosomes is said to be euploid. It applies to both haploid and as well as diploid condition, because that organism will have one complete set of chromosome and such organisms are called as euploids. Suppose if there is a variation, they are known by different names. One is aneuploidy. Aneuploidy is the variation in the number of individual chromosomes. There may be changes in the number of individual chromosomes. But if you look into the total set of chromosomes, there is no change. Because in each set there are two chromosomes. In one set, there may be one chromosome may be missing. Instead of two chromosomes or two sister chromatids, there will be only one. That is what you call aneuploidy, variation in the number of individual chromosomes. So this aneuploidy was noticed in 1916 when the scientist Bridges discovered X0 male and XXY female Drosophila. What do you mean by this? Aneuploidy here in 1916, the Bridges discovered the male and female. Male and female was having X0 chromosome, it was known as male. But whereas the female, it should have XY. but there was two copies of X and one copy of Y. There is an increase totally. As a result, what has happened in this? Instead of 46 chromosome, there is only 47 chromosome. But in earlier case, X0 condition, instead of 46, there will be only 45 chromosomes. The female Drosophila, which was having either 7 or 9 chromosomes respectively. Otherwise, in a normal condition, it should have 8 chromosomes, either one less or one extra. That is observed in 1916 by Bridges. Then this uh, aneuploidy, we have four more categories. One is nullisomy, monosomy, trisomy, and tetrasomy. Now let's try to understand this. Nullisomy, loss of one homologous chromosomal pair. These types are more common in the plants. Even human beings have also contributed to this. Nullisomy means loss of one homologous chromosome pair. In that monosomy, loss of single chromosome, loss of single chromosome, 2n minus 1. Then like monosomy, there is trisomy, one extra chromosome. This monosomy is very common in maize. Trisomy is in datura plant, which you can see. One extra chromosome will be there. Trisomic, see here, 2n plus 1. In the last there will be 3 chromosomes. Then in another condition, what you call tetrasomy, one extra chromosome pair. That means 2 chromosomes with one extra chromosome. That is what you call tetrasomy. So in the tetrasomy, there will be same thing, but there will be one extra chromosomal pair. Instead of 2 in a pair, in the last pair there may be 4. Now, how this happens? This is more common in the plants, and many of these things we have introduced also to get a better harvest. Triploidy generally results from diaspermy. It is the general result from dyspermia or dyspermia, diaspermia also. Fertilization of the egg by 2 sperms simultaneously. Very interesting. Generally it doesn't happen. Only one is able to enter into the egg. But by some means, if there is a fertilization of the egg by 2 sperms simultaneously, not only fertilization, if they survive to birth, even if they survive to birth, most of the time they die shortly after that. Most of the conditions, this triploidy is lethal. It will kill. So that's the reason why all chromosomes are present in equal number. It may be due to inactivation. How? The uh, here it will be lethal die since all chromosomes are present in equal numbers. Many of them may be due to X factor inactivation. There is a one sex cell due to X inactivation. Only a single X is active in the cell, not the double, but only a single X is active in the cell. So there's an imbalance between gene product from X and gene product from the autosomes. So autosomes and the autosomes are the body chromosomes or body cells. So why it is lethal? Maybe due to X inactivation. Here the gene X, which is meant for the sex of the individual, inactivation happens. Because here only single X becomes active in the cell, which may cause an imbalance between the gene product from the X chromosome and the gene products from the autosomes. Like triploidy, we have tetraploidy, four sets of chromosomes. Very rare and it is lethal. The animals or plant will die. This happens because sometimes there is a failure of first mitotic division. The first mitotic division, the cell will not be able to undertake. Before the commission of the first mitotic division, the chromosomes, they become very, this is a very rare condition and always lethal. First meiotic division fails, but the chromosomes replicate and divide. But all of them end up in the same nucleus. There will be two mitotic division. Where there is a failure of the first mitotic division, even if the first mitotic division has failed, chromosome again replicates and divide and end up in the same nucleus. Then another one is aneuploidy. Aneuploidy. So there are two causes for aneuploidy. One is non-disjunction. Just you remember in the cell division during the anaphase, the paired chromosome should separate and one group should move to one pole and another should move to the opposite pole. But suppose if the paired chromosomes, if they do not separate but they go to the same pole in the meiosis, when they move to the opposite poles as a result of the meiosis, there will be two groups. But now what has happened? Even a dividing cell having one cell having no chromosomes and another cell having all the chromosomes. This is non-disjunction, or sometimes it may be because of anaphase lag. In this condition, the chromosome move to the opposite pole so slowly that it doesn't get incorporated into the nucleus. when the nucleus are formed during the telophase. The cell cannot remain in anaphase for a undefined time range. But unfortunately here in this anaphase lag, the chromosome groups move to the opposite pole very slowly. Before they reach the opposite poles, the entire cell from anaphase enters to telophase. So as a result, what happens is, and after the telophase, there is a cytokinesis or division of the cytoplasm. And it gets incorporated into the nucleus as it forms a telophase. What is the effect of that? This effect is fairly well tolerated for sex chromosome. In sex chromosome, there is nothing much. But it is a bad news for autosomes. Why it is a bad news for autosomes? All autosomal monosomies and trisomies have been seen. But most are spontaneously aborted, but not here. So that's why there will be aneuploidy, because these uh, cells are not abundant. And if you talk about sex chromosome and aneuploidy, the variation in the chromosome number has different names to call. For example, here in a human being, there are 45 chromosomes plus one X chromosome. They are the normal, it looks normal. 45 plus X chromosome. So this is known as Turner syndrome, X0, because in the sex chromosome only one X factor is there. We don't know whether next one is a Y factor or the X factor only. Because this happens because of the absence of ovaries. That condition is known as Turner syndrome. What do you mean by Turner syndrome? All 44 chromosomes are okay. But as far as the sex chromosomes are concerned, there was only X chromosome. Another X chromosome or Y chromosome, it would have been missing. And that is known as Turner syndrome. What are the conditions? There is a folds of skin at the neck. Folds of skin. Don't compare it with the double chin. Even there is a more folds here. Short. People are short. Mouth always opened. Tongue trying to come out. Then even if you see the breast and nipples, they are widely spaced nipples. So this is what you call the Turner syndrome, which is because of the absence of one sex chromosome. There can be one more thing. The genome you see XXX and XXXX. These people are very tall, but they have menstrual irregularities. Even many times, or at least sometimes, they show reduced intelligence. They dumb people, reduced intelligence. But this is not been marked, not been marked. Usually, on they are found only mere by chance. Like that, we have one more XXY, what you call Klinefelter syndrome. Many times, he looks like a man, but he has uh, that's why we can call it the Klinefelter, feminized male, male but looks like a woman. May develop breasts. There can be elongation of the limbs. So they are discovered only by chance. Infertility is a common symptom among them even when they survive. There can also be another type, what you call XYY, tall, slightly retarded. Most of the time, either the people which are having XXY chromosome, they may be in prison or mental institution. Then the last type is YY without X. Because either it should be XY or XO or X0. Though they will survive up to embryonic state, and in the embryonic condition they become lethal. Suppose all these things, if it has started with a chromosome X, within few years uh, they could too many genes on the X but a very few on the Y. Otherwise, either it should be XY or X0. Instead of that, only Y, two Y chromosomes. Both are sex chromosomes. Both are same chromosomes. So such non-distinction can result in a person like 2X and a Y. As a result, what happened? 47 plus XXY. This is what you call Klinefelter syndrome. It's a male because that's a Y chromosome. Makes that person with Klinefelter as male. But he looks female body, hair pattern. Even there will be breast development. But they are sterile. Some developments may be delayed. There may be retardation. Unable to speak properly. Most of the time it has not been diagnosed. Probably for most of the treatment, we have to treat him with testosterone hormone. See here, Turner syndrome, there is only one X chromosome. In Turner's female, no ovaries will develop. So that's why they do not undergo any body changes during puberty and they are sterile. Hormone treatment can cure them, cure all, but not the sterility. So these are the conditions. I am not going to discuss about them. For the time being, we will let me stop here and continue in the next class because so probably for your information, next week ದಿಂದ ನಿಮ್ಮದು ಟೆಸ್ಟ್ ಸ್ಟಾರ್ಟ್ ಆಗುತ್ತೆ ಇಂಟರ್ನಲ್, and that's also MCQ. And I prefer to carry out those tests using Google Forms. Okay, thank you for attending.